live recording then. Yeah. Recording is also on. Recording which is
रिकॉर्डिंग नहीं शुरू हुआ है रिकॉर्डिंग अभी
last.
Dr. Deepti, have you started the recording?
Yes, ma'am. We have started the recording.
Okay.
Live YouTube.
And it is live YouTube as well.
Okay, fine, fine. We will start.
You got the TV with you, madam? Yeah. Should I go ahead with? तुम्हाला मिळालाय?
Good afternoon, all the delegates in the post-lunch session of the 32nd Quality Improvement Program, 4th day. After the lunch, our sugar spikes, and our next speaker is going to speak. The title of the topic is very interesting. The first part of the lecture is sugar cake. Sugar cake prevents memory impairment. So we already have sugar cake after the lunch, and let us see how the sugar cake helps. Dr. Santosh Rudravar has done his Bachelor's of Pharmacy from Amravati University, Maharashtra, way back in 95 to 99, and he has done his Master's of Science and Doctor of Philosophy from Naipur, Mohali. After that, he went for postdoctoral fellowship at Griffith University under Professor Mark. Then he did postdoctoral fellowship at University of Sydney under Professor Richard Payne. He was ARC DECRA fellow and lecturer at University of Sydney from 2014-15, and then he went to the industry. He was a group leader at Menzies Health Institute, Queensland, Griffith University in 2016, and from 2016 onwards, he's a senior lecturer at School of Pharmacy and Pharmacology, Griffith University, Australia, Queensland, Australia. He has some distinctions to his credit. He has Discovery Early Career Research Award, as well as the Australian government's funding of $4 lakh. He was the program director for Bachelor of Pharmacology and Toxicology. He has supervised many PhD and MPharm students. The patents and the publications at his credit are more than 45, and he has industrial research experience in discovery at Dr. Reddy's lab from 2001 to 2003. So I am waiting myself to listen to the first part of his lecture. Dr. Santosh Rudravar, please.
Thank you very much, ma'am, for your kind introduction. I would like to thank Professor Sharad Wakode sir for giving me this opportunity to present today. And I'm really excited to share my experience. I'm involved in both teaching and research. So today I'm going to talk about that, what I enjoy more, doing research and teaching. So, I can't see option here, share my screen.
You are able to share. You can share it. We already enabled you. Yeah, now you can do.
Dr. Santosh, are you able to share the screen?
Yes.
Yes, yes, yes.
Can you see my screen?
Yeah, we are
Yes.
able to see your screen. Kindly enlarge, maximize.
Yeah. This is okay now.
It's okay now?
Yeah.
Yeah.
Your face
Okay.
is only half where we are able to see. You can like lower down the. Yeah. Now it is perfect. Yeah.
Okay,
Right.
so yeah, let's start. So today I'm going to talk about my research and my teaching at Griffith University School of Pharmacy and Pharmacology. So this topic I have divided into two parts. First part I will talk about my research, and second part I will talk about my teaching and scholarship of teaching at Griffith University. So before we start, this is a workshop. I just want everyone's involvement in this. So uh, before I start talking about my research and teaching, I just want to know more about the participant, and then I can focus my topic and spend time on whether research or teaching. So at the top of screen, you can see there is a code there. So please can you use your mobile and go to menti.com and enter this code for me, please. So my first part is sugar cake prevents memory impairment, and the second one is advances in medicinal chemistry teaching in virtual environment. So before we start, could you please go to this menti.com and enter this code and answer this question? So menti.com, not Mentimeter. So menti.com and enter this. So after lunch, listening chemistry will is a bit challenging. So I just want to make sure everyone is awake.
It's again showing Mentimeter only mostly.
Yeah. So just menti.com. Don't go for Mentimeter. Menti.com. There should be menti. So Menti is for presenters. So for participant, it's a menti.com. So if you go to the menti.com and enter this code, if you're able to.
After going to menti.com and entering the code, what we see is the screen is coming Mentimeter, and in that the question comes, how are you feeling today?
Yes. So you just need to answer that question. So answer will appear here on the screen. So That's good. So we will start with this positive mood. That's great. So, okay, let's move to the next question. So that will give me some idea about from where you are and what you are doing. So just department, area in which you, if you are like, teaching, lecturer, which area you teaching, or if you're in industry, right. So second question. So same code. If you refresh,
मैम को कर दिया, मैं अनवर शेख सर को करती हूँ. अनवर शेख सर का 18-21 को है. Hello.
hello.
It is not
So,
going ahead. It remains at. How are you feeling today?
okay, it's not moving.
Yeah.
Is it clear what you wanted to tell, Dr.
Yeah. So able to see this screen. Is it same for everyone?
No. We can see your screen, but when we go to menti.com,
Yeah.
the presenter had changed slide. Go to the slide we have to make. Next slide.
Next slide. Yes, please, ma'am. Sorry
Now
about
we can see what is your job title.
yeah. So that question. So just understand the audience so that I can focus my talk.
Sir, all the audience is all our teachers.
Yeah, the teachers. Yes, ma'am. And most of them are like
are in the like
chemistry.
assistant professor to professor, rather associate professor they would be.
Okay, okay, that's good. That's great then. Okay, so thank you very much for your response. So give me a second. I will just connect. Can you see my slide now?
Yeah.
Sugar kick prevents memory impairment.
Memory impairment. Yes. Okay. Thank you. That's great. So when let's start. So when this human genome project was completed in 2003, so everyone was surprised to see a number of genes present in humans. At the beginning of this project, everyone was expecting humans will have more than 100,000 or close to 1 million genes because bacteria, E. coli got 4,000 genes, water flea got 30,000 genes, rice got more than 35,000 genes, and fruit fly got like 25,000 genes. So compared to these small organisms, so human, we are very complex, and everyone used to think that we might have more than 100,000 or million genes. But after the completion of this human genome project, and we have less than 25,000 genes. So what could be the reason for this? Everyone was surprised why we got so much less genes compared to other organisms. Is it like humans, we are surprisingly simple compared to these organisms? So the answer to this question is in post-translational modification. So post-translational modification involves modification of proteins with additional functional groups such as acetylation, phosphorylation, methylation, or disulfide formation, and glycosylation. So this post-translational modification means once protein is expressed, it is synthesized in our body. Okay.
Voice is not clear.
So, okay, sir. So once, is it clear, sir, now?
Yes, yes.
Yeah. Okay. So once protein is synthesized in our body, these proteins get modified. That's why we call it post-translational modification, and it involves addition of small molecules on the protein, or removing of those regulatory epitopes on groups from the protein. So various post-translational modification takes place in our body. Example is sulfation, acetylation, phosphorylation, methylation, and disulfide formation, and glycosylation. So these modification on protein makes this protein change its structure and function. And that's how we introduce diversity in proteome by this post-translational modification. So my group is interested in glycosylation, which is one of the post-translational modification. And glycosylation is addition of N-acetyl glucosamine shown in green color in this structure. So addition of this N-acetyl glucosamine on protein, which is called protein O-glyc acylation. So this addition on protein takes place at serine and threonine residue of previous proteins, and this is a dynamic process. So one protein can undergo addition of O-glyc residue multiple times during the life cycle of that protein. And there is another enzyme which removes that GLIC from this protein, which is OGA enzyme. So nature has selected this pair of enzymes, OGT and OGA, to do this O-glyc acylation or glycosylation on protein. So we can imagine, so OGT is like pencil, addition of this group here, and OGA is like eraser, so you remove this group. And by changing this addition and removal of this group, you can change the structure and function of that target protein. So far, more than 4,000 proteins have been identified as a substrate for O-glyc transferase. More than 4,000 proteins, which includes various cytoplasmic proteins, nuclear proteins, and mitochondrial proteins. It's a surprising that nature selected just these two enzymes to carry out this O-glyc acylation. Similar post-translation modification is phosphorylation, so addition of phosphate group, and with similar substrate set of substrate in human body, we have 518 kinase enzyme to add just phosphate groups on the protein, and we have close to 200 phosphatase enzyme to remove that phosphate groups from the protein. But why nature selected just two enzymes or just one enzyme to add sugar and one enzyme to remove sugar, that's still a mystery. And how come just single enzyme, O-glyc transferase, OGT, can modify such a wide range of substrates? So far, we have identified more than 4,000 proteins, which could be modified by O-glyc transferase. So that's why we are more interested in OGA and OGT, because it's a unique. So only one enzyme present in human body to this post-translational modification. So you can imagine it's straightforward that if you have any changes in this regulation of this glycosylation, it is associated with metabolic condition or disease condition. So it's not surprising that just a dysregulation of these two enzymes is not affected or not involved in any disease condition. So O-glycan transferase downregulation or upregulation is associated with so many diseases. Primarily, this O-glycan modification dysregulation is involved in cancer, neurodegenerative diseases, and diabetes. So my group is my group is more focused on neurodegenerative disease and cancer related to O-glycan transferase. So recently we summarized this. There are a range of oncogenic factors which get glycosylated and glycosylated by O-glycan transferase. O-glycan transferase is present in every cell, but is overexpressed in various types of cancer, solid tumors, and range of cancers. And it has been observed that the role of O-glycan in these is glycosylation of various oncogenic factors. So we are trying to understand, and we are involved in development of O-glycan transferase inhibitor probes, so we can understand how come a single enzyme can modify such a wide range of substrate, and what is the role of O-glycan transferase in cancer or stabilization of various oncogenic factors. So topic of today's talk is more focused on neurodegenerative diseases. So as I mentioned, a range of proteins, they get glycosylated or get modified by this O-glycan transferase enzyme, and it forms this glycoprotein. So one protein can glycosylated at several places on various serine and threonine residue. And it has been observed that, so there are various substrate proteins which are involved in neurodegenerative diseases. They are also get glycosylated with glycan residue. So which involves like amyloid beta protein, tau protein, alpha synuclein protein. So all these proteins in healthy brain get glycosylated. They have sugar on them. So once you remove the sugar, maybe by the action of O-glycan hydrolase, OGA, and then this protein can misfold and it forms this aggregation or precipitation of these proteins, and we call this, which is characteristics of Alzheimer's disease, tauopathies, and Parkinson's disease. So this glycan residue, this sugar shown in blue color, acts as a grease so that these proteins, they cannot clump together and they cannot form aggregate. So that's how it protects sugar. This sugar protects these proteins from aggregation, and aggregation of proteins is the main characteristics of this Alzheimer's disease or Parkinson's disease or various neurodegenerative diseases. So we are interested in what is the mechanism of this sugar residue, how it helps to prevent aggregation of these proteins which are involved in neurodegenerative diseases. So this recently Merck they reported on compound MK8719, which is a O-glycan transferase inhibitor. So this compound is in phase 2 clinical trial, and this is the only compound from this series. So what happens, this tau protein, which is involved in microtubule assembly. So this tau protein has both the glycan as well as phosphate group, and this happens in a healthy brain. So due to the action of OGA, and it removes this sugar residue on this tau protein. And then once you remove the sugar protein on tau protein, and then we have various kinases, they add this phosphate group on the tau protein. It forms this oligomer, which then aggregate to form PHF and neurofibrillary tangles. And these are the characteristics of neurodegenerative diseases. So what happens once this oligomers form, these oligomers, they are neurotoxic species. Once these oligomers forms, they kill the neuron, and then they aggregate and form this NFT. And when we observe changes in patient, like patient is having dementia, and then we characterize the patient is having Alzheimer's or Parkinson's disease. So in this process, formation of oligomer to NFT, it takes 20 years. So the damage, that's the formation of this neurotoxic species, takes place at a very early stage. So we do not have any diagnostics tool to characterize this oligomer formation, but we characterize formation of NFT as disease condition. So which is really wrong, because the damage has already done. So that's why we are more interested in identifying the role of this sugar residue in glycosylation and how it prevents formation of oligomers. So this MK8719 is the Merck compound, and if you see how this compound was developed, so the PUGNAC was the starting point for this OGA inhibitor discovery, which is a oxazoline transition state analog. Further modification on this part, making this cyclize, gave this thiazoline, which is selective, whereas PUGNAC is not selective, because GLICNAC is substrate for a range of glycosyl transferase enzyme. Then further modification on this, addition of this ethyl chain gives this N-both GT, so which is both selective and potent. And then addition of this nitrogen provide a thiamate G, which is both potent, selective, and metabolically stable. And MK8719, by replacing this hydroxyl group, which makes this compound lipophilic for blood-brain permeability, and this is the clinical candidate, and which is having both potency, selectivity, metabolic stability, and brain exposure. So this compound, recently they published phase 1 clinical results, and which is positive. And this is the first compound, small molecule, is developed for Alzheimer's disease or dementia or neurodegenerative diseases. So you can see that none of the compound worked in clinical trials so far, because this process is very complicated, and we target only this late stage of these neurodegenerative diseases. So that's why this compound targets the early stage. So as I mentioned, we are interested in identifying the role of GLICNAC, that's a sugar residue, in aggregation of various proteins involved in neurodegenerative diseases. So if you see the tau, neurofilaments, and alpha-synuclein, so these three proteins which are involved in neurodegenerative diseases, they get glycosylated directly by O-GLICNAC, and amyloid beta and Huntington's protein get glycosylated by various upstream mediators get glycosylated. So we identified, we'll focus on alpha-synuclein and tau for our research to identify the role of glycosylation in protein aggregation. And so far we know that there are few glycosylation sites which are mapped in alpha-synuclein, and this NAC segment from amino acid 61 to 95 is important for the formation of aggregation or structure of this alpha-synuclein. Whereas this tau protein, there are very few glycosylation sites has been mapped so far, and this serine 400 is important residue for aggregation. And again, amyloid beta, this amyloid beta 42 unit from 597 to 638 is involved in aggregation of amyloid peptide. So first we initiated this project with alpha-synuclein. Our hypothesis was, so when this alpha-synuclein is glycosylated by O-glycination, like addition of GLICNAC sugar, and that's what happens in healthy brain. So if you remove this sugar, this alpha-synuclein without sugar, and then it get aggregated and forms like involved in Parkinson's disease. So we thought, if we take NAG segment peptide and add sugar on that, and is it possible for this glycopeptide, the synthetic peptide, glycopeptide, to bind to alpha-synuclein selectively, binding to monomer, and inhibits this aggregation to polymer or this aggregates. So that was our hypothesis. So based on this, we prepared range of peptides and glycopeptides. So these are peptides and these are glycopeptides with this glycinac sugar. We also used L-amino acid as well as D-amino acid, and we used some linker as well as a glycine so that it can have desired conformation. So we follow this solid phase peptide synthesis method for synthesis of these glycopeptides. So this is a very standard method for preparation of peptides on a solid support. And for those who are new to solid phase peptide synthesis, I will just briefly discuss what exactly happens in a solid phase peptide synthesis. So first we start with a linker. So based on nature of your final compound, what exactly you want on the C terminal of peptide, carboxylic acid or amide. So then you select appropriate resin as a starting point. So you use this resin and then couple that resin with amino acid, an orthogonally protected amino acid. So the alpha amino group of amino acid is protected with FMOC, which is base labile, and side chain of the amino acid protected the acid labile protecting group. So you couple this carboxylic acid to the resin, you form first, attach first amino acid, then you deprotect this amino group, FMOC group from the amino group, and forms this peptide where free amino acid is there, keeping intact side chain protecting groups, which are acid labile. Then you follow the same coupling condition, attach second amino acid, again orthogonally protected, and then remove this amino group and attach third amino acid. So you repeat this procedure till you assemble desired length of peptide on solid support. So we followed this procedure. So this procedure was first reported by Merrifield in 1968, and he prepared one enzyme 369 124 amino acid containing enzyme, and he developed this this method, and for which he received Nobel Prize in chemistry. So we followed this solid phase peptide synthesis method and prepared these peptides and glycopeptides. And then we next tested these compounds whether they are inhibiting aggregation of NAC segment of the alpha synuclein protein. So we followed this THT fluorescence assay, which is the basic assay to test aggregation inhibition. And we use this natural product EGCG as a control for this, and we use alpha synuclein as a negative control. So we noticed that the compound 5 having this glycine linker has shown maximum aggregation inhibition result. So compound 5 was the best compound from this series. And then further to confirm validation of this fibril formation, we carried out blot assay, and you can see that this compound confirmed that this compound has significantly inhibited alpha synuclein aggregation. So there is a definitely role of this sugar and in inhibition of this aggregation. So now this is the preliminary results we published recently, and now we are working on understanding the mechanism. So now we are changing the aggregation parameters. So we are more interested in the kinetics. So how this aggregation inhibition takes place. So those things are currently we are investigating. Okay, so now move to another protein which is involved in neurodegenerative diseases, which is tau protein. So these are the few glycosylation sites which are mapped for the tau, and so this tau 400 it's very important. So there's a paper published in Journal of Molecular Biology, and what they mention. So if you add just one glycinac on this 400 serine residue of the tau, and it can inhibit this glycosylation. You can see that. So the black line is tau and the red line is glycosylation or addition of just one sugar residue
on this tau protein, which is a 441 amino acid containing protein. So you can see that just addition of one single residue can make a huge difference in aggregation of this tau. So without GLICNAC, this is a black line, and with GLICNAC, this is a red line. So it definitely inhibits aggregation. So based on this, we thought instead of using 441 amino acid containing peptide, can we use small peptide containing residues present in this 400 amino acid, maybe 10 to 11 amino acid containing peptide, not like huge protein. And we know that this 400, yes, serine 400 is important for aggregation. Like it plays an important role in aggregation. So addition of sugar at that position and that segment close to 400 is crucial for tau's aggregation. So based on that, we prepared a couple of peptides, glycopeptides. So we prepared this compound 1 as a standard without sugar, and then we prepared this 5 different peptides with sugar at different positions. So we moved this position of sugar. So to understand what's the role is this sugar is playing in inhibiting aggregation like whole protein, the tau protein. So we investigated this with tau protein, but still we are optimizing assay conditions for this aggregation. It's very tricky to deal with tau protein, so that's why we are struggling to screen these compounds. So there is no method available for screening of these tau compounds. Small molecules it's available, but not for the peptides or glycopeptides. So that's why we are still optimizing conditions for tau. Meanwhile, we thought what we test these compounds for aggregation of amyloid beta, which is also involved in Alzheimer's disease compared to tau, and more focus for research is always amyloid. So very few people they work on tau. So we thought these assay conditions are easy to reproduce and easy to optimize. So meanwhile, while optimizing our assay conditions for tau, we screen compounds for amyloid beta, and we surprised to see that compound 2 is really helping to inhibit aggregation. So this is a compound 2, and showing the red, and which is really good in inhibiting aggregation of amyloid beta. So this is the first example of tau derived peptide can also inhibit aggregation of amyloid beta protein. So we further carried out TEM images to show that this compound significantly inhibits aggregation of amyloids. You can see that 1 without sugar and 2 is with sugar. You can see that there's a significant difference in aggregation of amyloid protein. We further carried out Congo red staining, and this is the with compound 2, which is glycosylated peptide. So to summarize our findings, we can see that GLICNAC, a small sugar molecule, prevent aggregation of these proteins which are involved in neurodegenerative diseases. So GLICNAC acts as a grease and prevents misfolding and aggregation of proteins which are involved in neurodegenerative diseases. So what I will do now, I will move to the next part of my talk, talking about scholarship of teaching and learning at the School of Pharmacy and Griffith University. So what I'm doing in teaching innovation.
Good help to the faculty members as well as some their research scholars also. It will be helpful how to reach to the Griffith University. Please go ahead.
Okay, thank you, sir. So student retention and success is always our aim when we teach at university. So we focused on these 3 points: innovative unit content, active learning, and teaching with technology. And we noticed that if you use these 3 tools, definitely it will help for student success. So first I'll start with innovative unit content. So this is a very common picture in our classes. Students, they don't engage with what we teach, especially if it's a medicinal chemistry, and then definitely they are not interested in learning chemistry of these drugs. So we need to change this to this. So they should involve in writing structures of chemistry compounds, and they should involve in a discussion during the class. So we can help them. So they need to climb their own Mount Everest, but we can be their Sherpa to help them to reach or climb their Mount Everest. So for example, this is the example of innovative unit content. So if you are teaching chemical equilibrium, molecular reaction, equilibrium dissociation constant, and association constant, and if you discuss this chemistry like equation, and they might not show any interest in their learning. So if we try to correlate that chemistry equation with like, for example, KD of two drugs, losartan KD is 350 nanometer and nanomolar, and candestin is 7 nanomolar, and how it affects the dose of that compound, which is like 100 milligram and 16 milligram, and how it involved in half-life of this drug, like it's 5 minutes and another is 60 minutes. So if we try to correlate these equations and with the real life examples, and then they might show some interest, and why these like equations, they are important for understanding these physicochemical properties of pharmacokinetics and pharmacodynamics of these drugs. So now I'll focus on another example of active learning. So if you see this learning pyramid, so generally they get students get 5% during their lecture when they attend lecture. Then after lecture, if they do reading, they get 10%. If they watch something, 20%. If they demonstrate, you demonstrate them, get 30%. When they discuss in lecture and workshops, or when they practice or apply, and when they explain to their friends, they get the maximum. So we need to try to aim to get into this discussion like 50 to 90% of learning. This is average learning. So for that, we need to adopt some active learning strategies. So if you see this Bloom's taxonomy, so remembering as at the bottom of this triangle, this pyramid. So we need to at least push them for analyzing, evaluating, and applying various concepts they learn. So for example, if you teach them what are different functional groups, alcohol, ketone, their like general structure, their examples, they may not show any interest in these groups because it's quite boring. So if you discuss these simple functional groups and ask them, find out the interesting bits in these various functional groups in various drug molecules, and then they might show some interest. And that's what we notice, like, if you give this worksheet to them and say, identify functional groups, and then they correlate, okay, so these functional groups, they play a very important role in making up action of these drugs. So that's how they can develop their interest. So this is the first year B Pharm chemistry lecture slide. So scaffolding is very important for them to develop their own understanding. So we can provide information such as Lewis structure, molecular shape, molecular polarity, and intermolecular forces. So if you provide all this information to them, and that help them understand the pharmacokinetics and pharmacodynamics of these drug molecules. So this is another slide from intermolecular forces. So this is the example of captopril, lisinopril, and their IC50 values, and what are different interactions, hydrogen bond interaction, electrostatic interaction takes place with this carboxylic acid and this lysine, this amino group, primary amino group. So this Menti thing is not working. So that's what generally I do. So after every 5 to 10 minutes of lecture discussion, and we use this Menti, and they use their mobile to answer this question, and that's very important, because nowadays most of the students they use mobile, and always they have mobile in their hand. So we can't take away that mobile from them. So why not ask them to use mobile to get involved in discussion? And most of the time students, they don't answer questions because they feel like they are not right, and it's obvious. So if we use this tool menti.com, so they can participate in discussion, they can answer various questions based on this. So that way I find it very interesting. So we can get them involved in discussion. So after every 10 to 15 minutes of like discussion or teaching, so I will get them involved in this in-class activity for further discussion. Okay, so this is another slide, cardiovascular structure-based therapeutic evaluation case study. So Canada and US, they use this technique to teach medicinal chemistry. So for example, how drug 1 and 2, they are used for this patient, and how drug 3 option is more important or more helpful to this patient. So by understanding the structure-based therapeutic evaluation means you need to understand the clinical history of that patient, so to make therapeutic evaluations. Only by understanding the chemistry behind this drug, like every drug molecules contains some biological message in them, and you can only decipher that message if you understand all these parameters, such as patient history, other drugs involved, and nature chemistry of the drug. So this is another example. So this is why chemistry is important. So if you want to help and make therapeutic decision, for example, captopril and lisinopril, both are angiotensin converting enzyme inhibitors. So which one you will select for these patient condition? So definitely due to SH group, it might react with the food present in that food G-tube. So definitely you will not consider captopril for this patient, and lisinopril is the right option selecting here. But nowhere this thing is mentioned there. But if they know the chemistry of these drugs, so they can easily suggest the right option. So now talking about teaching with technology. So technology is really helpful if you integrate this technology in teaching. So that's what we did. So we tried to use technology. So now we are having on campus or online, blended learning, integrated e-learning. So many options are there. But if you integrate these technologies and techniques with innovative content, so you can really make student engaging lecture or workshop. So for this one of the course we teach in third year Bachelor of Pharmacy course, and we use this innovative unit content, active learning, and teaching with technology, both like 3 strategies to deliver a course. So this is based on this principle. So active learning. So what I hear, I forget. What I see, I remember. And what I do, I understand. So if they get involved in doing things, definitely they will understand, rather than if they just listen, they won't get it. So mixed mode of course delivery. So this is like we thought like medicinal chemistry content. It's delivered best if we make online lecture, like online videos, and they can access. It gives them flexibility. They can access this video at any time, and active learning. So then face-to-face workshop followed by video lectures, so they can get involved in discussion. So lectures, high quality video lectures, and workshops are face-to-face workshops. Okay, so as I mentioned, it's very important to integrate what you are doing, and appropriate integration is important for a maximum outcome for this exercise. So before we deliver this course, I prepared this activity map. So it shows what is their responsibility and what is our responsibility, and what content they are getting face to face, which content they are focusing on online. So we have like 12 weeks of trimester. So every Friday, I upload lecture notes and workshop questions on Learning at Griffith, which is a platform we use at Griffith. So every Friday, I provide them lecture notes and workshop questions. So every Monday, I upload mini video lectures. So all those video lectures are maximum 15 to 18 minutes long videos, not long videos, only short videos based on each drug class or clinical class of those drugs, which I will upload every like 6 to 8 videos every Monday. And then they analyze this, go through these lecture videos and lecture notes and workshop questions, try to work on those workshop questions. And every Friday we have face-to-face workshop, which we discuss these workshop questions. And at the end of this workshop, we have online activity. So that helped me understand what they are doing, how they are doing, and also that it will help students also to clear their misconception. Also, I can understand how they are doing with this. So this pattern we follow for 12 weeks. So this is the activity map. Every this thing happens. So this is a screenshot of one of the video lecture. So I use iPad, OBS, Air Server, a couple of these softwares to create these videos in my office. And we thought like if simple using PowerPoint slide, they will not understand what I'm talking about. So if they are not engaged in the chemistry lectures. So that helps me to write some notes on these slides and they can easily understand this. So this is another slide from this lecture, antibiotic. So that helps them to understand the chemistry when I'm talking about metabolism of chloramphenicol. So,
excuse me, sir.
So this is a video. We'll see whether you can.
So only modification allowed is on this R group. So you can have a variable R group. So the most important part, first one is beta-lactam ring, second one is carboxylic acid, third is cis hydrogens at this position, 5 and 6 positions, and amide substitution at this 6 position. So these are the 4 essential structural features of beta-lactam antibiotics or penicillin analogs, which is important for their biological activity.
So in this way, we use these 3 strategies, innovative unit content, active learning, and teaching with technology to deliver a medicinal chemistry content to pharmacy students. So this is another video from our school. So it's regarding therapeutics. So
welcome to the proton pump inhibitor simulator.
An incredible new way to learn.
We're not just rote learning answers, we're out there, we're making mistakes. Simulated setting rather than just throwing us straight towards a normal person.
It's such an incredible experience. It was really high tech, pictures were super clear, and interact, you can ask questions without causing harm, but you're learning and you're applying what you get.
With the pharmacy setting, it works really well. For a traditional classroom, learning like this, it brings it to life.
They're like real patients. There's a bit of sense of humour put in there.
Sincerely hope I'm not pregnant, seeing as I am a man.
But it was great. It was accurate. It's how somebody would be if you asked a man if he was pregnant.
High enthusiasm, great interest, excitement about the profession. The feel of technology creeping into pharmacy.
That's really cool.
So yeah, that's the example of how we can teach medicinal chemistry and therapeutics. So we try to use technology in most of the teaching at School of Pharmacy in Griffith. So this like doing teaching, it really helped. And this was I have planned not for COVID. So this is I'm doing from last 3 years. So this is my 4th year I am using this mixed mode technology. I never teach face-to-face lectures, so just these videos, and those videos are really powerful for them to understand chemistry. We got very good feedback, and now we are planning to writing manuscript to publish this innovation in medicinal chemistry teaching. So this was not at all related to COVID, but we planned just 3 to 4 years before. Like 4 years ago, I started doing lecture this. And it's not like that only if you're interested in teaching or research you do it. I'm more interested in research than teaching, but always we think we don't have enough time to do research. But I find if you do something in an innovative way, definitely you will find more time for your research. So I thought selecting this topic, both researchers and those involved in teaching might be interesting to them. Thank you very much. I'm happy to answer any question.
Yeah. Now the session is open for any queries by the delegates. Thank you, Dr. Santosh. It was very nice to listen to you. Long gap.
Yes, sir.
That is a good thing. You are here with us. Hello.
Sir, can I ask a question?
Yes, please.
Myself, Chitra Gupta from Lloyd Institute of Management and Technology. Just out of curiosity, I want to ask one thing. Do you suggest increased your findings from the first part of your lecture that sugar kick prevents memory impairment, and you suggested that sugar is important in preventing the neurodegenerative disorders?
Yeah.
So most of the neurodegenerative disorders happen as we age. They are more associated with aging.
Yes.
So do you suggest, did you have any findings pertaining to it that sugar intake be increased as one ages? Will it have
No,
any impact?
absolutely. No, absolutely not.
No,
This is just the just the catchy title I gave to this presentation.
Right.
Yes,
Okay,
yes. So
okay,
you should not consume.
Yeah,
you should not consume
that's
more
what I
sugar.
wanted to clear.
No. But sugar is important. Sugar is important.
Right,
So what happens when you say that most of the neurodegenerative diseases happens in old age? So that's related to sugar metabolism. When I say that all these proteins which are aggregated in brain, so in healthy brain we have sugar on those proteins, and in Alzheimer's brain or Parkinson's brain we do not have sugar on those proteins. So what happens? So due to the metabolic changes, like for example diabetes is the main cause of that. So due to diabetes our brain is not getting enough sugar, so there is a disturbed sugar metabolism in our brain. So that affects glycosylation of those proteins, and so that will lead to the formation of these monomers. And these monomer species and dimer species of these proteins, they are toxic, they are neurotoxic. And when you see that someone is forgetting things, means having dementia, so the actual dementia for that patient, the person, happened 20 years before you see the patient.
Symptoms.
So you see the symptoms. So the actual like precise diagnosis is postmortem. that's the only diagnosis we have for Alzheimer's or dementia. So that's not possible. Like,
Right.
so this thing happens 20 years before you see the symptoms. So that's why now everyone is focusing on early events of these aggregations. And that's why there is no drug available in the market from last 15 or 20 years. So there's no drug for these diseases because it's such a complex thing. So what happens 20 years ago, once they kill that neuron, it's gone. So you cannot modify, you cannot repair. So so this is just the catchy title. So
Right, sir. Right, sir. Right.
yeah, yeah.
Thank you so much.
Thank you.
Hello, sir. May I ask one question?
Yes. Yes, sure.
I want to know what were the problems you faced during peptide synthesis. That means its isolation and purification regarding.
So we didn't face any problem in isolation or purification. They're like simple protein peptides. So only we faced problem when there was sugar. We didn't face any problem. But when there was no sugar on these peptides, for example, NAC segment or tau 400 segment, it was very challenging to purify those peptide sequences because those are like aggregation prone sequences. So it was always precipitating. Even like 1 milligram of compound dissolved in 20 ml of solvent or acetonitrile water, it was very challenging because those are the proteins, the sequences which are in aggregation. So we notice that we can, like, without doing any assay, you can see that addition of just one sugar, it can make a huge difference, and those compounds can easily go in solution. So definitely we face problem, but then we still use sonication, maybe use very dilute sample to do HPLC.
Okay. Any particular synthetic procedure for this purification? That means.
No. We use HPLC, preparatory HPLC, and we use different columns. So instead of using C18 column, we use C6 columns or C8 columns.
Okay.
Yeah. So, and large pore size columns. So that's what we initially struggled with. C18, couldn't get anything out, and then we changed column to C8, C6, C4, and large pore size.
That means you made a combinatorial chemistry type synthesis like that. You added one by one amino acids, and then you get the peptides and you isolate it like that.
Yes, that's correct. So we added one, like, we did manually. We didn't use like a synthesizer, a peptide synthesizer. We used manually. We did manually, and yeah. So we added one sequence and checked confirmed formation of peptide after addition of 5 amino acids. We cleave the amino acid and cleave the peptide from the resin and analyze. So we just monitor reaction after attaching 5 amino acid, and then we did HPLC, LCMS, and purified those compounds by HPLC.
Okay. Thank you, sir.
Thank you.
Hello.
Hello.
Santosh, sir.
Yes.
Sir, can you throw some light on the physicochemical properties required for such molecule which you have designed, that is MK? What were the physicochemical properties you kept in mind?
So if you see this this compound, yeah. So this is not from my lab. So this is Merck's compound. So I'm we are working on this, but still we are working on non-sugar compounds. So we are replacing sugar in this oxygen with carbocyclic compound, and we send this compound to Merck for screening, and that's why I am associated with this. But for development of MK8719, I am not at all associated. But I am interested. So we have published this viewpoint in Journal of Medicinal Chemistry on development of this drug molecule. So here we can see that they have replaced this hydroxyl group with difluoro compound. So definitely lipophilicity is key for this compound to target the blood-brain barrier. But then shape of molecule is important. So we tried to first make some benzothiazole, but they don't form the same shape, so that didn't work. So now we turn to carbocyclic, similar to Tamiflu and Relinza. So we replaced the serine oxygen with the cyclohexane, but then chemistry with cyclohexane is a bit tricky. So yeah, I think definitely physicochemical properties and shape that molecule is very important.
Thank you, sir.
May I ask a question?
Yes.
I would study would help later to target the target blood system for
I can't get that. It wasn't clear.
No, your study would certainly help for a formulator to target medicine or target a drug to transform into a drug delivery system.
No, I can't hear you properly. I'm losing you.
Yeah, Dr. Rajesh, kindly loudly you just repeat your question.
Yeah, Dr. Santosh, I just want to ask whether your study in industry would certainly help a formulator to target a drug for treatment of Alzheimer's disease.
No, we are unable to listen again. Okay, so you can chat these things in chat box if you want.
Yeah, or you can email me or you can contact me.
Sharad.
Yeah, Dr. Sagar, please welcome.
Sharad, I think he's asking any formulation technology will enable to deliver the drug in the brain. I think that is the question.
Santosh. Dr. Santosh.
Yeah, so regarding OGA inhibitors, so yeah, so definitely that will help, but still I think still people have tried liposomes and nanoparticles, but that didn't work. So I'm not sure how that thing will work for this one.
So in a way,
So, but
Dr. Santosh, looks like there is no technology yet found to deliver drug into brain, right? That's what.
yes.
Okay.
Yeah, that's correct. Yeah.
Thank you so much, sir. Yeah. I think that's what I heard. It was very difficult, but he was talking about
Difficult.
what technology.
Yeah, it can. Yeah, you can use the formulation to just cross the blood-brain barrier, but then again going into the brain cells, and that's a bit challenging.
Yeah, crossing is possible, but getting further deeper, like you need chemistry other than that.
Exactly, and that's what this Merck's compound is about, and that's the only compound in the class.
Putting difluoro on that compound, usually when you put chlorofluoro, iodobromo, all this fluorinating agent, usually it end up in super potent molecule. Did you find a further stronger potency on those with difluoro?
No, not. But I think the problem with this one was like, if you see these all these compounds are potent. But only problem was selectivity, and then they overcame then the stability, and then finally they worked on that, and then stability, brain permeability, and then they worked with this fluorine. They prepared close to 1,000 compounds, and so this difluoro is the most potent compound. They do have some other compounds they mentioned, but then those other compounds have some issues with like metabolism. So their metabolites are toxic. The compound is active, but then metabolites are toxic. So when they changed other two fluoro groups as well, and those were the most potent compound, but then it formed this toxic metabolite. So they said, okay, we don't need that much potency, but this much potency is sufficient so that it's not having any other issues.
And that's a really good point, because when you put this fluorinated, either metabolite or compound itself ended up in higher half-life, or the toxic metabolite or compound itself.
Yeah. So they got like picomolar activity, and they say, oh, then if activity is good, but then liability is also there. So that's.
Thank you.
Very. Thank you. There are so many comments from Dr. Vittal Chopde from Modern College of Pharmacy, Pune. Excellent session, very valuable, useful information for PG, PhDs also. Then Spansh Shah has also mentioned the importance, what you have mentioned regarding the sugar. Then Manoj Shindeji also mentioned nice lecture. Milind Kate, other faculty members. Devrao Awariji, he has also mentioned very nice lecture. And to attend your special lecture, I think from industry also your friend has came to us.
I can see Vivek.
Vivekananda Malurwar.
Yeah, I can see him after.
Can you please?
After 25 years.
Yeah. When we are forwarding the things to various groups, it's a very good thing that people from across the India are joining, and you are just listening to know what is said in the USA.
Yeah, yesterday. Yeah. It was very impressive. I thought I will write him email about stuff.
Much interested to listen to you also, and next session is there with Sanjay Paisa, Umesh Paisa. So that is why he has also joined.
Yeah.
Malurwar sir, are you able to listen to us, Dr. Malurwar? He's from industry. He has.
Right now, am I audible now?
Yes. Some noise is breaking.
Yeah.
No, it's clear.
I would extend thanks to this get together with Dr. Santosh for a very long time.
Yes, more than 25 years.
Very fortunate that Dr. Santosh is my student. 25 years, almost 23 years
20,
before
yeah.
Classes at Vidya Bharati College, Amravati. After that, he joined NIPER, and it was very good future for him. And with hard work, what I have mentioned earlier, also last lecture, Dr. Sudhakar was there. So same way, Dr. Santosh has also done a wonderful career by his own hard work and sincerity, and that is why he is there in the Griffith University. Few years back, we had a chance to listen to him personally in DIPSAR for our students also. So Griffith University has given him a charge like as an ambassador to that university from India, so that he can help the students from India to join the Griffith University if they are having any problem or something like that. So in future also, if anybody wants, he can be of some help to all of you for your PG, PhD students also. He will be always helpful. My students are also taking help of him. So thank you very much, Dr. Santosh. So
thank you, sir. Thank you for this opportunity. I really enjoy it. That's great. Thank you.
Thank you very much. So as because of some technical problem, I think Madam Parley is unable to rejoin here again. But with
Vakode sir, I'm just joining.
Yes.
Hold for a minute, sir. I'll display certificate. Just I'm joining.
Yeah, that is what I was thinking. So people or the students can join for PhD also in your university, Santosh?
Yes, sir. We have scholarship for students. So there are currently 3 Indian students. They are working in my group, and 2 more they have recently submitted their application like last week. So we have PhD scholarship and also Bachelor of Pharmacy and Master of Pharmacy as well. So Master of Pharmacy, 90% our students are from India. Yeah.
Good thing, definitely to.
Yeah. So I saw most of the, I was lurking with the presentation yesterday and today as well. So they're more interested in doing research, and I was listening this morning and yesterday as well, doing research and patent and how competitive. Do you have 2 minutes? I can just say something about that. I share my experience.
Okay, madam wanted to share the certificate.
Okay.
You'll have
Okay.
to
Okay.
share
Okay.
that certificate.
Yeah. That's fine.
Next resource person is also waiting.
Waiting. Okay. That's fine. That's fine. Another time.
Then we are observing here. Milin Kate from Patal Dhamal Vadwani College also joined here.
Okay.
Hello, sir. How are you?
Yeah, good. Thanks. How are you? Nice to see you after
I
so
am
many
fine,
years.
sir. After I think so, 22 years after.
Yeah. After 22 years.
In between,
How
I
are you
am
doing?
find out to you, sir, but I'm not getting your number. मैंने बहुत सारे
Okay.
even Anand Paul sir, I asked him for your number, but he is not having. I think so.
Dr. Santosh, you have
तो
to
मुझे आपको देख के पूरे कॉलेज के दिन याद आ गए मुझे अपने. Still I remember जो आपकी जगह थी वहाँ पे मैं एक बार गया था जहाँ पे आपकी रूम थी वहाँ सब एक बार.
Dr. Bhayati has joined, I think.
Okay.
College of Pharmacy. Dr. Jagdi, are you able to?
Yes, sir. Thank you.
Yeah. Please welcome. We invited him for the resource person, but because of some problem with him, some technical reasons, he could not accept our invitation. Thank you, Dr. Bhayati. Thank you very much.
Thank you, sir. Thank you so much. Thank you. I would have been fortunate if I could have delivered something. But anyway, next time definitely I will try. It will be my.
Dr. Bhayati has completed the form from Amravati.
Amravati. Yeah. Hello, sir. How are you?
From Gujarat, PhD also. From
Yes. I'm fine. I'm fine. How are you?
Yeah, good. Thanks.
doing very nicely research in the pharmacognosy.
Hi, Santosh.
Yeah.
Involved in the organizing various conferences, seminars in Nagpur region. So very good thing.
Sir, we should organize one Zoom meeting for all Vidya
so
Bharati students if you have contact.
Remove your screen, this screen sharing, so that madam will be able to.
Okay.
Stop screen sharing. Yeah.
Hello, Santosh. I can see Santosh also.
Okay, that's good. Hello, sir. So,
Hi, Dr. Santosh,
I'll.
after a long
Yeah,
time,
good. Thanks.
almost
20.
more than 20 years.
20 years. Yeah. So I'll get your good. Thanks. I'll get your details from Wakode sir, and we should organize one Zoom meeting with all Vidya Bharati students.
Sure.
Yeah.
Certainly.
Because of the COVID also, portfolio, and because of the technology like WhatsApp and all the Facebook and the other things.
It's possible now.
Brochure is circulated, so the groups are forwarding to next groups, and teachers all over India are joining. That's a good thing. Welcome. Thank you.
That's good.
Madam, please. Yeah, thank you.
So, Dr. Santosh, very well done. And the way you have explained about the peptide synthesis, and you gimmicked us with your title. Everybody was, caught up in the title, and you were successful in gimmicking us.
So recently I published in Journal of Medicinal Chemistry, and they say, "Oh, your title doesn't look nice. Get some catchy or sexy title." And so that's what we came up with.
True. There was a paper which I published on Ratrani, and the title which I gave, the Nightingale. The Nightingale night. So something like that you have to do for the
yeah, exactly.
Good. And it was indeed catchy title. And you presented also very well, especially the peptide synthesis. And so your certificate for the day is displayed, which we will be mailing to you. Are you all able
thank
to
you.
see the certificate?
Yeah, I can see the certificate. Thank you very much, ma'am. Thank you very much.
So
That's
normally
great.
we
Thank
present in person along with a bouquet of flower, but this time we have to do it as a soft screen, So
no, that.
Here is your bouquet of flower. Hope you like it. These are nice purple orchids.
Okay.
Very nice.
Thank you, ma'am. Thank you very much.
Thank you, sir. Thank you for sparing the time. And
no, I enjoyed, and it was great pleasure. It was a great pleasure to see Wakode sir after so many years, and I feel proud to present.
And it is really wonderful the way you teach the students. The teaching methodology adopted by you people, giving them actual practical knowledge is creditable. Because generally it is one-way teaching which happens, and instead of one-way teaching, you are involving the students. That is something great.
Yeah, that's good.
So
thank you very much.
Thank you so much for coming and sparing the time. What's the time there now? Around 7 o'clock?
8 o'clock, 4.5 hours ahead. Yeah, 8 o'clock, 8 p.m.
Yeah. Right. You are at Sydney or Queensland itself?
Queensland.
Queensland, right.
Yeah,
right.
Gold Coast.
Right. The picture is so beautiful.
Yeah, that's very close to our, you're 4 kilometers from our university. So that's why you never see students in my class, So that's why 4 years ago, I decided to if you're teaching on Gold Coast, they don't come to the university. So you need to provide some options to them, and university have recorded, everything is recorded. So they say, we'll listen your recording. Why don't need to come there? It's not come to university. And that's why 4 years ago, I thought how I can get involved and engage these students. So that's why we created, but chemistry, if you just do PowerPoint and talk chemistry,
no, it is
don't
not
understand.
possible.
Yeah. So today also I wanted to use iPad and show how can I use this one, but couldn't connect it. Maybe this internet and Zoom thing is not matching.
But because of internet and Zoom, at least we could bring you to these QIP delegates. So that is a blessing in disguise.
Yes, guys.
The COVID-19 one way is a blessing, because you all people could join us.
Yeah, absolutely. Yeah.
Even virtually, but that is okay.
Yeah.
So next time, do plan to visit our institute. It would be
pleasure
to have you.
Yeah, sure, ma'am. Sure.
Right.
Thank you very much.
Sir. Thank you.
Thank you, sir.
Thank you, Dr. Santosh, and thank you,
sir.
Faculty members, Dr. Bhayati, Dr. Koshish Gabne,
Mallurwar sir. Awari is also there. I saw somewhere Awari joining.
Madam, go ahead. Dr. Pai, Mr. Pai.
He's there?
Yeah, join.
Yeah. Okay. Our next speaker is Dr. Umesh Pai, who is Associate Vice President, Head of R&D and MSTG at Sun Pharma USA. Umesh has completed his B.Pharm from College of Pharmacy, Nasik, way back in 1993, and Masters from University Department of Chemical Technology, UDCT, Mumbai, which is ICT now. He started his career as a research associate in product development with Sun Pharma Advanced Research Center in India. In 1999, Umesh was transferred to Sun Pharma USA, and since then, he is working with Sun Pharma group of companies over the past 25 years. It is said that rolling stone gathers no mass, but it looks that Dr. Umesh Pai has started with Sun Pharma and rose to the associate vice president level, so proving this, saying wrong. In his current role, Umesh is the head of R&D and MSTG, that is Manufacturing Science and Technology Group, for solid oral products. Umesh leads his team for product and analytical development, method validation of generic drug formulations, for filing ANDA, addressing FDA queries, and pre-approval inspections. He also leads process validation, cleaning validation, and product lifecycle management activities. He is responsible for launch of new products to the market and life cycle management products such as change of source for active drug substances and excipients, batch site changes, and product site transfers. Among his professional activities, Umesh has provided various talks at AICHE, Marcus Evans, and IFPAC events on QBD, that is quality by design, and NIR technology. He has co-authored white paper with PQRI and currently is part of quality working group for AAM. I would request Umesh sir to explain, because though I'm reading, I'm not following what is PQRI and AAM. In his personal life, he loves to spend time with his family, cook new recipes, listen to music, and teach photography. Great. Great biodata. Dr. Umesh Pai is not only the vice president there, but looks to have an all-round personality at home, as well as at play, as well as at the work. So it is really nice to have you, sir. I'm unable to see, sir. Sir, yes. Is your video on, sir?
Yes.
Yeah.
First, thank you for the kind introduction. And also I would like to thank Professor Sharad Wakode for extending this invite for speaking and sharing my experience. PQRI is Product Quality Research Institute, and AIM is our institution for all generics. It's for Association of Accessible Medicine. And I would speak some aspects of it during the talk today. So we can start. I will share my screen. I will share my screen.
Yeah.
Are you able to see my screen?
Yeah. Yes, we can see. Okay. Maximize it, sir, please.
I will do it. I'll do it. Okay. So that's the talk we are going to have today. Generic drug products, facts, impact, and development. So thanks to DPSRU University for this opportunity again. And like I said, been doing this for 25 years, worked in the Indian market earlier before I got transferred to US. Have got to work with various dosage forms, injectables, solid orals now, semi-solids before, liquid orals, dispersion systems, a very small stint with transdermals as well. So I was very lucky, I would say, to get work, develop, launch a lot of these dosage forms in my career. And before I begin, I would like to say these thoughts are mine. It doesn't present the position of Sun Pharma, and it does not replace the details presented in all the regulatory guidances and independent professional judgment. So before I tell you what's the outline today, I would like to first thank all the teaching fraternity who is on the call. Because of all of you and the efforts which you all do, we are able to continue doing this effort, working in this industry. So a humble salute to all of you for doing this in and out. Teaching is very tough. It's very tough because just for preparing one presentation, I know what it takes. In order to deliver one lecture, and you are doing this day in and day out. So great effort from all of you. And before I say anything on generics, First, I would like to thank all the fraternity and whoever is present from the brand industry. I see Dr. Sudhakar Garad is also online. Thanks to all the innovation done by the brand industry. And what work generics industry does not replace what the brand does. So, but if I look at generics, it is a different world altogether. When I say a different world altogether, meaning there is an objective to reach the common man in terms of having medicine at an affordable price, and that's what our objective is. And when I say to the common man, there is a slide which will help you understand, at least from the economic impact point of view, at least in the United States, what I mean. So we will briefly discuss what is generic drugs. If you have already had such sessions in the past, so I would please pardon me if things are repetitive. I would present some recent facts which I collected about generic drugs. We will look at the economic impact of what it makes, only specific to the United States. I was not able to gather data for the entire world economy. How are these products developed? And what are we taking away from this time which we are investing? What are we taking away with us? So that is going to be the outline of the talk. So when I say generic drugs, I would read out the definition to you. So these are medications which have the same active ingredient as the brand and yield the same therapeutic effect. So key to remember, there are two key milestones for the generic drug industry, which we have to remember. One is in 1984, two senators from the Congress in United States, they established what is known as the Hatch-Waxman Act. It is the Drug Price Control and Patent Term Restoration Act, which established the approval pathway for generic drug products. So before 1984, there were only brand drugs in the world, right? And this act enabled pharmaceutical companies to file an application to the FDA seeking approval through what is called the abbreviated new drug application, or the ANDA. What brand companies filed is a new drug application, which is much more elaborate in terms of what is studied in terms of its safety and efficacy. So safety and efficacy is something which the brand companies establish, and we generic companies do not have to establish. And the second most important milestone for the generic industries is in 2007, there was the GIVE initiative. So that's a generic initiative for value and economy efficiency, which helped modernize and streamline the drug approval process for generics, and various other generic dosage forms could be introduced in the market. So those are the two important years to know. So when we say generic drugs, what does it actually mean? I just pulled out two labels from the Daily Med, right? Let's look at a brand name product label. Lipitor. This is used for reducing cholesterol. If you see, Lipitor is the brand name. It is registered. You can see that with the registered trademark. The drug is atorvastatin calcium tablets. The dose is 10 milligram. It also displays that it is a prescription-only medicine. It is a bottle. That's why it has 90 tablets inside of a bottle. The label displays how it is stored, where it is made, what is the expiration date. Now let's look at a generic drug product label. So I picked up a label from one of our owns. So this is atorvastatin calcium. The dose is same.
Can you just
same count.
loudly?
Huh?
Thoda loud. We need.
Okay, better now?
Yeah, it is better.
Okay, let me increase the volume. Is this better?
Yeah.
Yeah, thank you. So if you look at the generic drug label, it's the same chemical compound. It is same dose, same count of tablets, same storage conditions. So except for the name Lipitor, everything is the same. So let's look at some facts. So when we say facts, let's start from what is the commonality between generics and the brand. So as we've seen from the label, it is the active ingredient, which is same. The safety is the same. The effectiveness is the same. The strength is the same. The quality is the same because it goes through the same rigors of the approval process as of the brand. And it has the same benefits. So when we say quality, what it entails it has to pass the same quality standard as the brand name version, meaning the US FDA goes through the same rigorous review process, and that's how the generics get approval. And the FDA ensures that the generics provide the same clinical benefit and effective and safe as the brand. They can look different, right? When a brand is formulated, while the generics is formulated, the brand might be covered by patents. And when you come up with a formulation which has the same ability to have the same effect, you could be different. But that is also governed by guidances now through the size and shape guidance. You cannot be so much different than the brand, but there are limitations in terms of the size and the shape as well. So there are allowable differences, but it does not impact how the generic works. But even though they look different, they are very safe and effective. So in terms of cost, like I said before, they cost a lot of less money as compared to the brand because we don't have to repeat any animal and clinical studies to demonstrate safety and effectiveness. So also, brand is launched by one company. Generics, there could be multiple. And as you know, when there is more supply, right, there is competition. And as per the competition, the cost comes down. So it comes to a point where the generics cost so low and cost up to 85 to 90% less than the brand. So I would also like to give some examples. The competition here is so fierce that some of the pharmacy chains owned by Walmart, they would give some of the medication free without even have to pay anything, especially medicine for chronic disease management, hypertension, diabetes. Walmart gives the medicine free to certain club members who have a subscription for doing shopping at Walmart. They would give them free. That is the level of fierce competition for generics. So all this data is on the FDA website. I would share the references in the end. So any questions so far? Is this voice okay, Professor Wakude?
Yeah.
So I would say, please stop me in between if you have any questions. I would love to have an interactive session as well. So no formalities, all right? Now let's look at the economic impact. And this is only pertaining to United States. In last year, the total business of generic drugs was $115 billion, which is 8,62,500 crores. So that was the total generic business in United States. If we look at the annual prescriptions dispensed, 90% of them have been generics, 10% of them have been brands. But in terms of cost, the generic cost only 22% of the total drug cost. So when you might be wondering how this data is calculated, because in United States, when it is a prescription medicine, the pharmacy you cannot just go to a pharmacy store and buy what you need. It is very controlled here. You need to have a prescription from the doctor. You have to fill the prescription into the pharmacy and you are dispensed and everything is computerized. So at the end of the year, there is a record which is easily generated and all this analysis can be done. So a year before, the data is not available for 2019 yet, 4 billion prescriptions of generic itself were filled. The average drug copay, meaning when you buy a prescription, based on your insurance, you have to give some money to get your supply of medicine, correct? It could be a 30-day supply, 90-day supply. So that would cost the consumer here about $5.50. But the average cost for a brand would cost somewhere, if I round it up, to $41. So there is further analysis. So 95% of the generic prescriptions are filled at $20 or less. And the savings have been enormous. In 2018 itself, it is around $300 billion. But in the last 10 years, it is almost $2 trillion. So now why is this important? Because if we look at any population across any country or the world, you will see there are a lot of economically poor sections of the society who also have health conditions. So objective is also to keep them healthy, right? As a pharmaceutical fraternity, our goal should be to keep the entire humanity safe and healthy, right? Make the medicine reachable to all of them and have it accessible. So this is the work I would say is done by generics. Once, thanks to all the brand companies who launch and all these beautiful medications year and year after improving their profiles, right? We make it reach to the other part of the society as well. So key point is, how is this developed? So in the line of your program from AICT, which is the quality improvement program or whatever new things you are looking at research, I would just like to touch base on how these products are developed. And that is very interesting to see how these products are developed, because this development methodologies which have been adopted now in the generics are very interesting. And before I start talking about development, I would like to tell you all these resources are available on the FDA website. Everything is available to everyone. If you go to www.fda.gov, you will see the drugs at FDA. You will see what are the approved drugs. You will see what are the therapeutic equivalents and the approved drugs in the so-called Orange Book. You will see product-specific guidances for developing generic drugs. You will see a list of who are the first generic drugs who have been approved. If you are developing a complex generic drug product like transdermals or extended release injectables, the FDA has a pre-ANDA program where you could interact with the FDA, have the program discussion with them so they could guide the generics to develop safe medication. The Orange Book also lists the patents from the brand company, then exclusivities, then which are the approved generics. Then they also publish the inactive ingredient database. We cannot just pick up anything and make another formulation. We have to pick only certain excipients from the inactive ingredient database and only to a certain levels. And last but not the least, there are numerous guidances on how to develop the product. And the key is the ICH Q8 based quality by design. So when I say quality by design, many people don't know where this concept started. Who is the guru of quality by design? So the guru of quality by design is Joseph Juran. He's a Romanian who migrated to United States. He was an engineer, and he was the one who started the concept of quality by design. That's his book, J.M. Juran. So there is a Juran trilogy, what they say, and he is born in the early part of the 20th century, right? And you can imagine 100 years back how the quality was being perceived. And if we look at how they are perceiving quality, it is very great indeed. See how it is perceived. Quality, as per J.M. Juran, has two meanings. First, there are presence of features which create customer satisfaction. And second, not only presence, the reliability of these features, meaning ability to produce the same quality again and again. That's reliability. Any failures in these features would create a customer dissatisfaction, right? So removing the failure is the purpose of quality improvement, but creating the feature which is essential in your product is the purpose of quality. So nicely explained. This is like almost so many years back. This concept was first adopted by the automotive industry before it was adopted by the pharmaceutical industry. Now the entire pharmaceutical industry has adopted QBD. Even the brand medication companies, generic medication companies.
Umesh, sorry to interrupt you.
Yes.
Are we talking about the Toyota model in this?
Yeah, Toyota model, he was a consultant for Toyota as well. Yes.
Yeah, this is more on the lines of the Six Sigma thing.
Exactly. Yes.
Lean management, perhaps.
Yes. Thank you, Samgram.
So sorry to interrupt you.
No worries. Good. Yeah, it was a nice break for me also for talking. So guys, please pitch in and let's keep it interactive, right? So, thank you. So how did FDA do it? FDA collaborated with European Medicinal Agency, and based on the ICH, they created a lot of guidances, correct? And these are the guidances which the FDA and the entire regulatory fraternity has created. And especially if I look at ICH guidelines, guidelines Q8 to Q11 are key guidances for referring to when it comes to the whole process. So Q8, the second version, talks about the formulation development. Q9 talks about quality risk management. Q10 talks about the pharmaceutical quality systems and how to govern all this process and keep it reliable, like we said. Q11 is now talks about the active pharmacological substances and biologicals. Q12 is under implementation right now. It talks about life cycle management, and Q13 is in works. It is the continuous pharmaceutical manufacturing. Q12 would be the analytical quality by design. So the expectation is not only you should have your formulation developed through the quality by design methodology, but also your methods. So that is the latest. So if I look at the definition of QBD, it's a systematic approach. And one key thing to remember is you start with the end goal in mind. That is very key to know. What is your goal? That is where you always have to go. So it's like when you go for any expedition, you always have a goal. So development in the pharma industry is like an expedition. It has to be planned. You have to know where you are going. What are your key points to achieve? Like when people climb Mount Everest, right? Their goal is the summit. And there are 3 base camps they have to achieve. Similarly, in development, there are milestones which we have to achieve. And we will touch base on those. So that's what quality is all about. So this is based on sound science. And the process is governed by the quality risk management. Like I said, predefined objectives, and it emphasizes product and process understanding, and not to say also process control. So let's start with the process, right? So when we say we are developing something, what are the key milestones in the process? So start with your goals, right? It's called the quality target product profile. Then you have to predetermine what quality attributes you want to design in your product. You have to perform a risk analysis to evaluate the risk of each component of your formulation on the finished drug product quality. Then you have to mitigate those risks. You have to mitigate the risks in your formulation, your process, and packaging. And then you have to design a control strategy. Remember what Juran said, reliability of those features. So this is how you establish reliability, through having a control strategy. So these steps are given in ICH Q8 R2. So quality target product profile. So it's a prospective summary characteristics of a drug product which you have to achieve in order to get the desired efficacy, safety, and quality, right? So it forms the basis of any development. It starts with the dosage form and type. I've put here an illustrative example of a tablet, right? Let's say we want to develop a tablet, ABC hydrochloride. The dose is 100 milligrams. So you have to specify what you are developing, meaning you are developing a tablet. It is this salt of this molecule ABC. What is the dose you are going to develop? 100 milligrams. This has to be very clear. What is the type of release? There are various release mechanisms one can design in a pharmaceutical dosage form. Immediate release, extended release, delayed release, pulse release. So for our discussion's sake, let's keep it immediate release. The route of administration has to be clear. It is oral. What is the shelf life you are targeting? Is it going to be 2 years, 3 years? So people start with 2 years. And what is your goal of pharmacokinetics? You are going to be bioequivalent. What parameters you will be targeting? These are given in the guidance for any specific products. Like for ADHD management, the guidance is more elaborate. It's not only Cmax, area under the curve, 0 to T and 0 to infinity. They also have partial AUCs. And as far as quality attributes is concerned, what are the key quality attributes which you would be targeting? Dissolution, how the drug releases, what is the assay, what are the limits for my dose? Is my drug a narrow therapeutic index molecule? So what should be my specification? That range has to be targeted based on the therapeutic index of a molecule. The uniformity, the related compounds, elemental impurities, microbiology. So those are the quality attributes you target. How are you going to present it in the market? Is it going to be a unit dose pack like the blister drug product, or is it going to be a bottle pack? So all these are the targets you set and decide as a team, along with the cross-functional team when you are given the task of developing a product. So once this is done, the next step is you have to list down all the predetermined quality attributes and you have to predetermine them. Yeah. And especially identify in all these attributes, which are going to be your critical ones. So description, how is my product going to look? How I'm going to identify it? Is it going to have any specific size, shape? What's going to be the assay, related compounds, and all the targets, whatever you put in the front, you have to sit and write them down. And lately, it's the nitrosamine impurities also, along with the elemental impurity. You see for valsartan, there all the people had to pull back the product from the market because of nitrosamine impurities which were formed while synthesizing the API. These are very cancerous, and the latest is metformin group, where while synthesizing metformin, they also found nitrosamine impurities. Now we have to take a look at, when we design a new product, is nitrosamine impurity generation possible during my API synthesis and the API which I'm using? That makes it critical to understand upfront. What is critical for the stability of this product? The assay, the related compounds, drug release, and micro. What is critical for bioequivalence in my product? How is my drug release going to be? we have to have the drug release similar to brand, and it is governed by statistics of similarity factor. And once this is done, we do a risk analysis. So when I say risk analysis is done to evaluate the impact on the finished drug product quality. Your risk could be from your API, your formulation components, your process, your packaging components, and all this is governed through quality risk management. Let me give you an example here, an analogy, which will help you understand. Let's say we want to cross a street or a road, right? If we are standing here and want to cross to the other side of the street, you know what we are doing? We are doing a risk analysis. We are looking at the traffic. Oh, there goes a scooter, there goes a car, there goes a truck. If a truck is coming, would we cross the road? No, that's your major risk. That's a high risk. If a car comes, that's a medium risk, because if the car hits you, you could break your leg. You may not lose a life. If a scooter hits you, you would get injured, you would fall. It's a low risk. So what is the risk mitigation we do when we cross a road? We wait until the traffic stops, and then we cross the road, correct? So that way we take care of the low risk, medium risk, and high risk. And we ensure that we cross the road and we have no risk at all. So this is the same analogy we have to keep in mind when we work with development, right? So let's start with active substance risk analysis. So the question is, if my active substance, if my dose is known and the safety and efficacy is established, what is the risk to me? Is the first question would come in many people's mind. But we have to see that the physico-chemical properties of the API are very key in order to understand the impact on the finished drug product, right? So what are the three key physico-chemical properties which could impact your assay, related compounds, content uniformity, dissolution, and stability? In my opinion, they are solubility, polymorphism, and the drug chemistry. So why is solubility is important? Yeah. So let's say the API is insoluble. So if it is insoluble, the particle size of the API is key. So how do you make it available for absorption in the body? So that is a knowledge which we discover. That is something which we discover when we develop the product. And these are the things which are not available in the common domain for the generics. So it is not easy to develop generic products, is what I'm trying to tell you. We have to first establish what class of the compound it belongs to. What is the biopharmaceutics classification system it belongs to? Is it BCS class 1? Is it BCS class 2? Class 3? Class 4? So that is why solubility is key. Solubility is determined in various pHs across the physiological range which is found when we take the medication. Polymorphism. When the drug is made, it could exhibit polymorphic tendencies. It could crystallize in a specific form which is stable, or it could change the crystal lattice as a function of time. And as the crystal lattice changes as a function of time, it could have various changes in its properties. It could have changes in its stability, and it could change the way it solubilizes, correct? And that could impact your formulation performance on stability. So polymorphism is key to look for. Chemistry. The drug has its own impurities. There are process impurities and there are degradants. So one has to understand under what conditions these degradations happen. Do they happen under oxidative conditions, acidic conditions, alkaline conditions, heat, light? So those are the studies which we do upfront because literature may not be available to that detail. All these studies help the analytical team. They do all these studies. It helps them develop proper sample preparation conditions, proper chromatographic conditions when they test the API, because the objective is also to see you don't degrade the API when you test the API, correct? That is the objective under which this is done, and also from a formulation perspective. So that is the risk from the active drug substance. What is the risk from the formulation? So when we say risk from a formulation, what goes in a formulation? Two things go in a formulation. Two class of things go in a formulation. Actives, excipients, correct? There is no, I'm not talking about the drug device combination. That is the third component. So here our talk is limited to only solid orals, right? We are not talking about drug device. So if I look at the formulation risk analysis, there are two areas, actives and excipients. So from a formulation perspective, if you know the BCS class, if you know the dose of your product, if you know the size of the tablet which you have to meet. What is key for us is the particle size, right? And what particle size is optimal for your targeted formulation is what you have to think. If the dose is high, the risk on uniformity would be relatively lower. But if the dose is, let's say, one of the products which we developed has a dose of 0.125 milligram. So when we say my tablet weight is 100 milligram and my dose is 0.125 milligram, that is a very small percentage. There would be a risk of content uniformity, right? We could have an issue in quality, and that is those are the things we have to think of upfront. Yes, as if my dose is so low, how I'm going to design my formulation? How will I mitigate my risk? What is the process I will choose so that my API is distributed uniformly in the tablet is what we have to look. Excipients. So what is the risk from excipients, right? When we put two things together, is it causing any incompatibility, right? Do they interact? Because this is coming so close when we compress into a tablet, they come so close that they could interact, right? We all know. And would the excipient selection impact its dissolution, and would it impact its clinical performance is what we have to see. So what are the risks from the process? From the process, when we say the risks, what are the risks on the assay, related compound, CU, dissolution, and stability? What is the process I'm choosing? And what is the technology which is suitable for my targeted formulation performance? We just discussed if the dose of the API is very low, we might have to think of granulation as one of the technologies rather than a direct compression if the dose is too low. So for the manufacturing process which is chosen, we have to perform a stepwise risk analysis to see what risks are coming up from each process and how they could impact the finished drug product critical quality attributes. So packaging, last but not the least, we would present the product in a pack, correct? And if the drug, is the drug sensitive to light? What is the desired pack which we are going to send to the market? Is it a blister pack with a transparent blister? Is it an opaque blister pack? Is it a bottle? So all those things, so those studies which you have to do under risk mitigation. So what is the risk mitigation process? We touched base on it while crossing the road, right? So as we said, we would cross the road when we see a green, when the traffic stops and we see a sign which says walk now, walk across your zebra crossing, the traffic is stopped. So what is the light and what is the traffic stoppage in the development? So it comes under the class of the formulation and analytical development. So all these risks are mitigated through drug excipient compatibility studies, studying the polymorph, studying the change of polymorph on stability. We use XRD to understand that. We optimize the formulation performance through multivariate design of experiments. We use statistical softwares, Minitabs. We develop stability indicating analytical methods. We develop discriminating dissolution method to see Do we have a matching drug release profile like the RLD? We do development stability studies to understand the risks of the product stability, and we assure the bioequivalence and stability performance. We would do pilot bio studies and understand what is the volunteer size we would need to conduct a final bio study, what design we should adopt for the bioequivalence study. Then in terms of process, we would do a processing design of experiments. We would take batches, we would do stress experiments, and we will see the impact on the critical quality attributes. Let's say you are blending something, we would over blend it and see what is the impact on the dissolution, what is the impact on uniformity. Packaging, we would place the product in stability in various packs, various counts, various types of packs. If the drug is sensitive to oxygen, is the drug sensitive to light? So based on that, we design all those packs and we conduct all these stability studies. That's how we mitigate all the risks. That is the key aspect of the development. Last but not the least, let's say we developed all this, completed our reports. How do we ensure that when we manufacture this in the plant, how do we control this and maintain the reliability? So that process is governed through the control strategy. We would have input material control, right? We would put specifications for actives, the excipients, all the critical aspects which we identified during the development, like particle size, the grade of the polymer, the type of the excipient. Like if it's extended release, there are various molecular weights of polymers, right? We could specify which molecular weight that, let's say hypromellose, we could specify it should be K4M, right? And that is the one which it will be tested to. Process controls. So when we govern how to make a product, it is governed through instructions, like manufacturing instructions through a batch manufacturing record, how to pack it through the packaging instructions. So those are governed through the instructions. At each stage, we would check, is the quality is being designed as we make it, right? So we would do some in-process checks. So one of the key things to see it and understand is use of ASTM standards, E2709, 2810, which gives the statistical confidence of passing the USP content uniformity test. So those are the tests we will build up in process to get that assurance. And last but not the least, we have finished product critical quality attributes put in a COFA and the stability specifications. So when I say critical quality attributes, those are in the specifications. We would build description, identification, assay, content uniformity, related compounds, residual solvents, microbiology, and with the limits. Everything comes with limits. So that would be designed. Future and some of the companies which have already adopted for in-process, designing an in-process specification is still a hybrid model of the traditional way of controlling quality. But if I look at NIR technology, that is something which gives you data in real time. That is probably a way to go for in the future for most of the pharma industry. But it is still in its infancy. Not many companies have adopted that technology. All this is governed through the ICH Q10. So once all this is developed, you have placed your control strategy. Now you have to tell the FDA that I'm going to make this product. Company XYZ is want to make this product. So you have to demonstrate now to the FDA that you are able to make this product, right? This demonstration happens through a batch, which is made in the manufacturing environment using qualified equipment, all these approved specifications and processes and validated methods to test. You have to demonstrate a batch, correct? So there are two key requirements when FDA looks at how you are going to make your batch. So under the manufacturing requirement, they want you to make the batch 3 times. not only one time. For, let's say your product has 1 strength, you'll have to make 3 batches of that strength. If there are 6 strengths, you have to make 18 batches of total. They are demonstrating 3 of each. You have to make the batch with the same formulation, which you are going to seek approval and propose. You are going to sell it in the market. It should be packed and manufactured in the same way you are going to make it in the future, right? The batch size is also governed. We cannot just make something on a desk, make few tablets enough for bioequivalence and stability and say I'm done. It has to have a certain criteria of a batch size. Minimum should be 100,000 units or 1 lakh, and or you could have a bigger batch size based on the market, foreseeable marketing demand. So we study the market before. Based on that, we design the batch size, because once it is approved, one can scale it up to 10 times without having a supplement to file and seek approval for the FDA during the life cycle management. So that's the batch size. And we have to make these batches using at least 2 lots of API. This was not true until 2012. Until 2012, we could make 1 batch of each drug product, each strength, with 1 active drug substance lot. Now that has changed. Now it is aligned with the EMA. So if you have to file a product in US or Europe, you have to take 3 batches of each strength and you have to use at least 2 different lots of API. Why? Because sometimes you might cherry pick a lot and say, I've done my job and here it is. And then realize when you start making this product, oh, I have a stability issue, I have a performance issue, I have a dissolution issue, and then start recalling product from the market. So the concept of QBD is learn first and not learn later, right? So that's why, and when you learn first, you demonstrate your learning through this mechanism. What is the testing requirement, right? So we have to use validated methods which are proven, and we have to use all the approved specification. Last but not the least, although we demonstrated stability during development, we have to demonstrate the stability on this batch which we just manufactured, right? We have to demonstrate stability at accelerated conditions. Now these are conditions for US, right? It could be different for different geographic zones in the world. So that is provided in the ICH guidance as well. So accelerated conditions, before we submit data, we have to have 6 months of stability at 40 degrees Celsius, 75% RH, and 6 months stability at long term as well. But before FDA gives a shelf life of 24 months, they would like to see the data of 12 months also. Only when we see significant changes on accelerated stability, we would also test at the intermediate conditions. Second important aspect before we get approval is the bioequivalence study. So all the bioequivalence studies which are required, there is a product specific guidance. It is given on the OGD website, right? So the generic products are considered bioequivalent with the innovator if the rate and extent of absorption are the same. And this has to be proven in healthy human volunteers as far as the generics are concerned, right? So we have to perform the stability, perform the bioequivalence study with healthy human volunteers. And what we would typically do is, typically all the AB rated drugs in the Orange Book, you have to conduct an in vivo pharmacokinetic study. Here we have to have criteria for Cmax, which measures the rate of drug absorption, and the extent of absorption is understood through the AUC, right? AUC 0 to T and AUC 0 to infinity. There are also in vitro methods for establishing bioequivalence. If the product is rated by FDA as AA, you don't require a bioequivalence study, pharmacokinetic bioequivalence study. You have to prove through dissolution that you are able to have similar rate of drug release And those methodologies are especially true for BCS class 1 molecules. And there could be probable molecules where instead of a bioequivalence, you can go through the BCS way of proving equivalence. One of the drugs which is there on the website has both these options, which you could prove the bioequivalence through the BCS class. Filing. So when we come to filing, correct, the Hatch-Waxman Act requires the ANDA holder to declare how it is going to file your ANDA, correct? Because one has to remember, when the brand industry does the innovation, they get a protection for their innovation. There are patents. The first patent is on the molecule itself. It's called NCE, new chemical entity, right? So when the product is launched by a brand and it is a new chemical entity, so those are protected for 5 years. We cannot have any ANDA submission for 5 years. So once the 5 years are completed, once the patent protection on the NCE is over, the FDA allows further extension of patent protection because they have lost, the brand has lost some time during the review, right? Yesterday, Dr. Garad explained when they file the patent, right? So we should give that protection to the brand, because if you look at the cost which is invested by the brand, it was averaging 1.5 to 5.5 billion for the brand industry to bring in one molecule, correct? For the generic industries, we have to remove 3 zeros. It is like 1.5 to 5.5 million dollars, right? Because we don't have to do the discovery and establish the safety and the efficacy. So that cost is saved there, right? But that innovation is protected. So when we file ANDA, there are 4 types of certifications one can do based on what are the patent certifications in the Orange Book. That means there is a product in the Orange Book where there are no patents listed. You can go through the Orange Book molecule by molecule. You will find there are certain molecules, there are no generics, but there are no patents listed as well. But those might be difficult for someone to make a generic, number one, or it could not be of any business interest for the generic industry. So next is paragraph 2, means somebody has made this product, the patents are expired over a long period of time. Now, not many generic companies are interested in this, but a new entrant might be interested in this product and they would file a paragraph 2. If the patent is strong, but then you would like to market it immediately upon patent expiration, one would file a paragraph 3 certification, right? Because in any of these 3 certifications, if you file, you will never have a litigation with the brand industry. But there is the most interesting category. It is called a paragraph 4 certification, right? When the NCE patent is expired, right? And when the brand can file once the NCE patent is expired, but there are certain patents which are still active. And that's where the paragraph 4 filers come. So the generic industry is incentivized for a first to file, and we will discuss that briefly in the next slide. And that is called a paragraph 4 certification. You would either invalidate that patent in the Orange Book, or you could say, you would argue it is not relevant. Yeah. So we discussed about the protection innovator is provided, the NCE exclusivity, and how it allows to extend the period of protection, right? And how the generic companies can file a paragraph 4. So interesting thing to know is, once a paragraph 4 is filed, within 45 days, the innovator industry has to sue. They put a lawsuit against the generic company that, you know what, you are infringing my patents. I'm going to stop you. Once there is a lawsuit, you get an automatic 30-month stay on the approval of the product. There is automatic 30-month stay, during which that is a period during which the litigation process happens. FDA cannot approve the product in those 30 months. But sometimes the innovator may not sue also. So those are the cases where the generic formulation has come up with an infringing formulation, where when the innovator looks at that formulation, because all the details have to be shared with the innovator. How is your formulation design? What is the mechanism? So the generic industry might have had a non-infringing formulation, and they would say, guys, if we have a lawsuit, we are not going to win this, and they may not sue at all. So that is another case. So I have seen a couple of these cases as well. So those are the interesting things which happen. So once all this is done, we have established, we have completed development, we have signed off the product development report, we have made the exhibit batches, we have established bioequivalence, we have established the stability. Now we file the product to the FDA, right? We have filed the ANDA. Now the interesting part is how the approval process happens, right? First and foremost is FDA has to accept your ANDA. So there is a checklist. Have you submitted each and every document required by the FDA guidance? So there is a check. And FDA could refuse to receive your application. The cost to file an ANDA today is $176,000 US. So once upon a time it was free, but now it comes at a cost based on the Generic Drug Fees User Act. So that is the fee. So that is a big investment for a generic company, right? So one has to do the homework, look at the entire list of requirements which are required to be filed for that ANDA, and then file. Once it is accepted for review, then the FDA would do a chemistry review, CMC review. Under CMC and microbiology, they would review everything. Analytical, stability, manufacturing controls, they would review everything and see that you have the ability and you have made the product and you have addressed, because the FDA is answerable to the consumer. Their objective is consumer safety, right? Although the objective is to have affordable medicine, but the objective is also to ensure that the medicine is safe and effective. They then review your bioequivalence. In how many volunteers did you establish your bioequivalence? Did you meet the guidance criteria? They would check your labeling. Are you labeling in line with the innovator? We cannot have a different claim than the brand. We cannot, because it's a generic. And then last but not the least, if the industry has filed a product which it has not been inspected for, FDA would go to the site and conduct a so-called pre-approval inspection. That also, the abbreviation is like my last name, Pai, right? So when people ask me, Umesh Pai, what has to do with pre-approval inspection? I said, no, I am a Pai from India. It has nothing to do with pre-approval inspection, right? So in the pre-approval inspection, FDA would come at, let's say you have a manufacturing site, you are making only tablets. And now you have filed a new ANDA, which is an extended release formulation based on pellets, right? FDA would come to the site, they would look at your equipment, they would look at your actual records, they would really see that you have made a batch. And second, they would see whether you have capability to manufacture the batch on a commercial level. Are you ready to give the product to the consumer if FDA approves you? So these are the things they would establish in a pre-approval inspection. If they have any feedback, they would share it with that firm. The local inspectors would do that. If there are any questions from the FDA reviewer sitting in DC, these people would verify and do the checks and balances and then give the feedback to the FDA reviewer. So that's what happens in the pre-approval inspection. And once all that is done, FDA would approve the generic drug application, right? So in fact, I told a lot of things. I would give a quick summary on the development, right, as a process. So it all starts with the design conceptualization, right? And you define your target profile, your critical quality attributes. You study all the regulations. So you make a strong case and make a mission statement, right? And you plan your mission. Then you do a systematic development. You based on all, yeah.
Umesh, apologies to disturb you. I need to log off. My day started already here.
Hey, no problem, Sudhakar. Thank you for joining.
Very informative, Umesh. Just to let you know, the audience, Umesh, myself, Sharad, we were colleagues together, and I had a great opportunity to have Umesh as a colleague during BFARM. Very friendly, very supportive, very caring. He would be always top. I never beat him. I was always second or third. But the friendship what we had was amazing. We never had a competition. It was very supportive. And that kind of behavior, the collaborative behavior from college when it starts, it stays all the way with your life. And that's why where he is today. And, so just keep in mind that among the faculty, among the professor, only thing you can carry is collaborative nature so that you help each other and take to the next step. So with that, I will take off. And Umesh, wonderful job. Thank you so much.
Hey, thank you, Sudhakar. Thank you for your kind words.
Thank you.
Yeah.
Take care.
Yeah.
Bye.
Bye.
Yeah.
Bye, Sharad. Bye.
Thank you. Bye. All the delegates must know that Umesh Pai was our topper for the Pune University. He used to always top in every classes also. There was competition from the education point of view, but as a friends, we were very close to each other every time. And as he has already said, Umesh, that he used to refer so many books, and that's why in his presentation also we are seeing the book reference of books and everything. So it's very nice to listen to Umesh. Please.
Yeah, sure. Thanks, Professor Wakude. So we do a systematic development because we have now conducted our risk analysis and we are mitigating all the risks through this process. Once we do this, our design is realized, our design is complete. Then we control it through the quality risk management process. Then we send it to the FDA to review and get the design approved. When I say design, I mean the formulation. And what we get in the end is an approved generic product, which is safe and effective and have developed a quality product, right? So takeaway, when I say takeaway, I would say, just remember this 4 things today from this lecture, only 4, nothing more. It is too much to learn and it's too much to grasp. And there are websites available for it. You don't have to remember each and everything. I just gave an overview of all of that. You can read and there you could also discover more details than I have done as well. But the main 4 takeaways I would love you to see is, one is generics possess same quality, safety, and efficacy as the brand. And that is a fact. That does not go away. Economically, there is a big reach. Hold on, let me go to my. Economically, there is a big reach to, in terms of reaching the common man as far as generics are concerned, correct? Because not all will be able to afford the cost of a brand medication. So that is the pious work which the generic industry does. As far as development goes, generics are developed by the same mechanism and methodology of a brand. It is done by quality by design, right? The objective is to have a bioequivalent product and a product with a stable shelf life. And last but not the least, the approval process, it goes through the same rigors of approval process as the brand. It is judged with the same quality standards. There are no different quality standards for generic, right? So those are the 4 takeaways I would like to remember from today's session. Yeah. So these are the 3 main references I had from my presentation. One is ICH guidelines, all the FDA guidances, and the Association of Accessible Medicines. It was called GPHA before, Generic Pharmaceutical Manufacturers Association. Now it is called Association for Accessible Medicines. So the name has changed. That ends my talk. That's my email. You could reach out to me, umesharpai@gmail.com. So thank you for patiently listening. In between, I see this text now. I was speaking maybe not in a loud voice, not enough to hear. Last but not the least, I am not a doctor. I saw you address me Dr. Umesh Pai. I could not complete my PhD. I had to join industry. My father passed away while I was in B Farm. So I had to take the responsibility of the family. Couldn't do a PhD. I could do masters because I got a scholarship. That's the only reason. Otherwise, I would have not done that as well. So that's my story. Thank you so much, all of you. I would be happy to take questions, all of you. Please feel free to ask questions.
Now the session is open for the delegates for any query or any comment you'd like to have. Madam.
Yeah. So I must congratulate the Trimurti. Trimurti is starting from our Dr. Sharad Vakode. Then we had Dr. Sudhakar Garad, and today we had Umesh Pai. So these are the three Murtis. I do not know them personally, but during the presentation, I just observed a common link which I would like to describe. All three are very humble. The three are connected some 25 years back, and still they are sparing time to attend each other's lectures, motivate each other. Dr. Sudhakar Garad, while leaving, commented that collaboration is the key to success, and all three are remaining collaborated with each other. So this, their behavioral aspects are giving you a lot along with their technical content. Do you agree, Dr. Pai?
Yeah. I'm not a doctor. Call me Umesh.
Actually, we
have gained in industry is more than that of the doctorate. What is there? Even madam, you are talking about 20. It's about 30 years old thing. 93 we finished B Farm. So not only 3, but all the 60 students which were there in the class are totally together till now. And one more person will be there in the last day session, Dr. Vikas. So he was again very nice person and a very really a real scientist from our class itself. There are so many others friends also joined here. Those are from the marketing field, some vice president and the big tycoon, business tycoon from the Thana. Mr. Sangram Joshi is also there. He is a very good leader in that part, pharmaceutical distributor throughout Thana. Can I see Sangram over here?
I saw him asking question
No,
in between.
Yes, it is a pleasure. It's a pleasure to listen to those three murtis, as Madam Amruta said so wonderfully. And I'm sure that you have said three murtis, but by the end of the session on the 19th, you will correct yourself to the 4 lions on the Ashok Stambha when Vikas Shirsat joins in.
Why not?
Tomorrow.
Even I can join Sangram Joshi to that. I would like to see you, sir. If you put on your video, I can see.
That will only be the Stambha, no Ashoka.
Sangram, that is.
Give me a minute. I'll just connect a video. Give me a minute.
Yeah.
Any other delegate wanted to discuss anything?
So there are a few questions in the chat box. One I see is how BCS classification would be beneficial to get biowaivers. If you study the solubility of a molecule and you determine it is highly soluble, and you see a guidance in the generic page saying that it requires bioequivalence study, what you have to do is you have to prove the permeability of the molecule. So the permeability of molecule is done through the caco cells. Those are the epithelial cells from the intestine, and there are companies who do this work of establishing permeation. Absorption systems is one of them. So if you establish permeation, and that permeation is established through known molecules which are not supposed to permeate and which are supposed to permeate through a caco cell, if you establish permeation, you can make a case for not performing a bio study and filing your ANDA with a BCS bio waiver. So that is possible. I see that question from Dr. Satish Polchettiwar. Thank you for asking this question. And FDA also realizes this, right? And FDA recently, for semi-solids, they also have now come with in vitro methodologies to prove bioequivalence. So those are in the semi-solid space. In terms of solid oral space, they are specially associated with BCS biowaiver based on this caco cells only as of now. Another question I see from Dr. Satish Polchettiwar, can we get biowaiver for lipid-based formulation or transdermal formulation? I worked briefly on transdermal formulation. After that, it was not continued at our side. That research still continues from our R&D in India. It is difficult to get biowaiver for transdermals. No, at least for all the prescription products which we got associated with, transdermal formulation, especially when I say transdermal, meaning patches, it is not there yet. For semi-solids, if you consider them as transdermal, yes, there is a in vitro methodology which is available.
Thank you.
Any other queries?
How US FDA do the inspection and how you solve the queries, from Dr. Gajanan Kalyankar. So when FDA comes at the site, it is called a pre-approval inspection. So two types of queries, FDA queries happen. Once the ANDA is filed, FDA would give queries called information request, right? Under the generic drug user fee, they would send questions. If they have any concerns about how we have done our studies, they would get clarifications or ask for additional data. And we would have to do it during the review process. Once that is done, like I said, if the facility has not proven itself or has not been inspected for that manufacturing that class of the product, FDA would visit the site. It is a surprise visit, right? They would not announce that they are coming for a pre-approval inspection. They would give a form 482 and say, we are here for conducting a pre-approval inspection, and that is our objective. So then we host the FDA. They sit at the site and they look at the entire ANDA. They ask questions specific questions as to how these batches were made. They would go to the facility. They would check logbooks and see whether we really made a batch. They would look at the MBRs. Let's say it is signed by a few people. They would ask to say, okay, get me this person in the room. I would like to interview this person. So we'll have to present the person, and the person there has to answer the questions directly to the FDA. So we cannot say anything in between. So those are the checks and balances FDA does. Once they establish that you have done all this work, you have answered all their queries. They also sometimes clarify any queries from Washington, meaning where the central FDA. And they would also look at your intended proposed manufacturing process. They would see, when you say, I will make a batch of 5 lakh tablets, I will use this blender, I will use this granulator. They would really go and see, do you have those equipment? have you qualified your equipment? Are your people trained to use those equipment? So those are the things which the FDA does when they come to the site for conducting a pre-approval inspection. So next question is, I see from Dr. Jagdish Baheti. Is bioequivalence study compulsory for all generic drugs in India and USA? In USA, there are product specific guidance. So if the guidance says you require a bio study, one has to do it. There are also products which do not require a bioequivalence guidance. Let me give you an example. If you Google Orange Book and you open and write the product methadone hydrochloride tablets, it is rated AA. It does not require a bioequivalence study. So it is not necessarily compulsory, right? Because as per FDA, even establishing a similar dissolution is like proving that your formulation would be bioequivalent only because your formulation is your API is a BCS class 1 molecule. That's what it means. In India, I'm not sure about India recently. That would be beyond my scope to answer your question. But I would try to get that answer to you. Please email me so that I would be able to answer your question from perspective of India, because that's where we do not develop the products for, but I have colleagues in R&D, right? So I'm only a part of R&D of Sun. Sun has 2 R&Ds in India, one based in Delhi and one based in Baroda. So they would know better. Next question is from Dr. Satish Polchettivar. Harmonization of all regulatory guidances from FDA and who needed. Could you comment about that? I am very much in agreement. What we see is when we develop a product for US, and let's say we want to file the same product to Canada, which is our neighboring country, or we have to file the same product to Europe, we again have to go through rigors of many documentation and all. It would help if there was one guidance, one requirement, one standard for the entire world. It would make the life easy. What I mean to say is, let's say if we prove if we make a generic and we have proven bioequivalence and versus the innovator, and every different country you want to file the product, you don't have to conduct the bioequivalence study again. Why? Because the FDA can work with the innovator and find out when you have launched in 10 different countries, do you have a same formulation launched in 10 different countries? If that assurance is obtained by the FDA from the innovator, they would know that. Then it would save a lot of the effort which is done by the generic industry in order to file generics in the other geographies. So that is what my comment is for your question. Yes, I am very much in support. We technically face challenges when we want to file products in different countries. Now, apart from filing products in US, my team, my team is 61 people here in US, right? And we file products for Canada, we file products for Mexico, then also we would be filing products for other countries. So these are the difficulties we face. So thank you for your question, Dr. Satish. Yeah.
Just need to log out of the screen sharing part, especially.
Okay, hold on.
Welcome, Sangram Joshi. Yeah.
Okay.
Welcome.
Hey, Sangram. How are you?
Yeah, it is nice to see the 4th, addition.
Yes.
And the way Dr. Vakode has said,
I will,
you are an entrepreneur.
Yes, I will tell you. For me, the highest regard goes to the teaching fraternity because of all what you do, we are what we are. Number one. Second, when it comes to working in the industry, in my opinion, there are only 2 classes of people. One is the entrepreneurs and there are the others. So no matter what we are in our department, we work for someone, right? So for me, even the entrepreneur is someone who has his own small stall of making a chai. For me, he's an entrepreneur. And for us, Sangram is that class of a person. So Sangram, we cannot do what you can do.
And there is a unique capability in an entrepreneur.
Yes, the risk-taking ability. I talked about risk
Ability,
mitigation,
and
right?
yes, it is not only risk-taking ability.
Yes.
It is calculated risk-taking ability.
Yes, yes, yes.
So
So that we don't have. Yeah.
So
that
we
makes
have
him
to,
unique.
you know,
Guys,
That makes him unique from all of us.
Yeah. And, you know, in between the, my description was, you know, cut short. All three people, they have come from the humble background, but that did not stop them from reaching the heights. So that is the clue you have to take, each one of you, irrespective of your, you know, economical background, irrespective of the support of the parents, irrespective of the problems which people face, still they grow because they want to grow. They have the desire to grow, and that requires the thought. What kind of thoughts you have, whether you have the thoughts which stop you or the thoughts which let you go ahead, because the thoughts are created in our own mind. No one can help that, because the thought, then attitude, then comes the action. Action leads to habit. Habit leads to character. Character leads to your success or failure. So this is all starts with a thought, and I think all the classmates, the way sir described, they have the good thought process, so they are growing. Now coming to Dr. Umesh Pai, first let me congratulate you for beautiful slides. Very simple, but very, you know, neat and tidy, going ahead with each point. So first for the slides. Second, of course, you have cleared the, you know, you have summarized the generics, how generics are equivalent, how economic impact is there of generics, how they have to undergo all QBD to prove the equivalence. So my question to you is, though you have said that generics are equivalent to branded drugs, now the point comes is, you have said that the prices are less. Now in India, another concept which we are discussing, I think 2 days back only, generic concept is coming where the cost is almost equivalent to branded. So then in that case, what is the use of the generics?
So the cost, if it is high for a generic, we have to understand how many people are in the field. Usually, like I told, when a generic, I will first start from the perspective here, right? When it's a first to file generic, who gets they get a 180-day exclusivity. So during the 180-day exclusivity, because of the efforts done by the generic, they are rewarded for a period of 6 months, where the price of the generic would be 85% of a brand. So only when the 180-day exclusivity completes and when the other generics come in the field, the price starts dropping, right? Because now there is competition. And if I'm selling my product as 85% cost of the brand, the second generic would come in and say, they make an agreement with the wholesalers. They would say, I'm selling this product at 75% of the cost. And then that particular group would start buying the medicine from the second generic. Then the third generic comes. And that is how the price erosion, it is called the phenomena of price erosion that happens. So in India, if you are having a high price, it could be two things. It is difficult to make generic, number one. For example, if I look at paracetamol, you will get it at few naya paises, right? Per tablet. I'm talking at a per tablet cost. But if you look at, let's say there may be a fentanyl transdermal patch which is applied in the hospital. So those might be costly. there might be complex injectables which are lipid based. So those are costly technologies. So the price of a generic is governed by two things: complexities and the competition. So when we know when it is complex, there won't be many players. So you will see every pharma company can make paracetamol, but can every pharma company make a lipid based injection? Probably they will be handful. And when they are handful, they know if the innovator is selling at, let's say, 10,000 rupees, I could sell it at 6,000 rupees as well. Because now they have done equal investment in terms of cost. But if you compare it with the brand, they would be probably lesser than the brand, always.
That is there. I was talking in this regard. I do not know the correct picture. I'm just trying to open up in front of everybody. I was just talking to one of the stockists, and super stockist he was. And he said bidding occurs for the generics.
Yes.
They decide the price. The stockist says that this is what I'm going to sell for. Maybe, and the profit margin sometimes is whooping 600%, 700%.
For the stockist.
For the stockist, reason being, say the bid is of 2 rupees and he sells it 40 rupees.
So that's something related to how
so
it is controlled.
That is where we have to,
I will tell you, if I may, the situation here, right? The US industry for generics is complex. So when we make medicine, we cannot sell it to Walmart directly, or we cannot sell it to any small pharmacist directly. This is associated, there is a complex network of insurance companies, the pharmacy benefit managers, and the wholesaler. The biggest, if you look at all the wholesalers in US, 3 wholesalers have capability to do wholesale business of 85% of the entire products in the entire United States. So McKesson, if you look at McKesson as a wholesaler, their revenue is bigger than Johnson & Johnson. So J&J is the biggest pharma company in terms of revenue. But if I look at McKesson, who is a wholesaler, has the biggest turnover compared to J&J. So now the question is, who is doing the hard work? The innovator is doing the hard work, doing the innovation. We are doing the hard work, trying to make it accessible to the common man. But who is reaping the benefits? It's the middleman. So that's true across the world. So between now in US, so there are various insurance companies. There is competition between them. So they would work with the pharmacy benefit manager, and they would say, in my formulary, if you have this insurance, you could get only this medication. So there is a competition there. All that is governed by cost. And that's how the marketing has to play in all these scenarios with everyone. And,
that is what, if McKinsey is the main player, they can always negotiate with Johnson & Johnson. At this cost, I'm going to purchase. Are you going to give or not? And then I'll sell at whatever price.
Yeah. Sangram would be able to give shed more light onto it from a perspective from India. I'm very away from the Indian market.
And right now, one of my student, the project which I had given was PG student, just recently submitted the thesis. I was having a doubt whether the government supply medicines are of the equal quality to the branded. And the study was taken up like this. You collect the 3 brands from the 3 generics from the government supply and similar brands from the market. But somehow, the outcome was both are equal. This project came into my mind because when I visited Baddi last year, the Polo Pharmaceuticals, these are so small pharmaceuticals, and we are talking about QBD and all, nothing is followed. Just nothing is followed, and still they are able to sign the contracts with the people and sell the products. And that is how I took this project. Anyone else? Any queries? Dr. Bakode, please unmute yourself.
Yeah. Sangram would like to comment on within 1 minute.
Yeah. First, the session is being recorded, so that's a difficult proposition. And yes, I do agree to some of the thoughts which Amrita Madam just mentioned about the way the generic market in India. First of all, we have to understand that the way the US market works and the way the Indian market works is absolutely chalk and cheese. So there's no similarity whatsoever, other than the thing that Lipitor contains 10 milligrams of atorvastatin, and Sorvas, a brand popularly known in India by Sun Pharma, Ranbaxy now, contains 10 milligram of atorvastatin. So other than the similarity of the basic drug being 10 milligram in the both the things, it's chalk and cheese. The US market, like we saw about in Umesh's slides, that the average price of the dollar price is between $5. Correct me, Umesh, if I'm wrong.
Yes, yes.
$5.06 you said, and pricing never exceeds about $20, the highest one. So whereas the Indian market is totally different. First of all, Indian market, as far as the insurance industry is concerned, is not governed at all by the OPD. In India, to get an insurance claim, you have to be admitted for 24 hours is the basic thing which goes wrong against the common man's accessibility to medicines. We talk about lot of changes, we talk about the drug price control order, we talk about essential medicines, we talk about NLEM, we talk about so many things, we talk about drug price control order, we talk. Unfortunately, till the time we have a GST on medicines, how can we even think of a fact that it is a common man thing? See, these are the basic fundamentals on which we have to talk sometimes, till the time there is a 12% GST, till the time there is a 18% GST, how can we say that we are trying to reach to the common man? What does the government want from us? As an industry, does it want to collect that tax which is so important, or make accessible use of medicines? So this is a different topic one again. Let's park it aside. Second thing which we talked about, Madam Amrita's question was whether generic products which are currently available, say for example a product is available at 2 rupees and some sells at 40 rupees. A common example of this is a brand called as nimesulide, which got the very much unfamous Kuvikyat molecule at the 2 rupees and 40 rupees bracket. Nowhere ever was the efficacy or the bioavailability of the nimesulide being compromised. I'm very happy that your own PhD students, PG students analysis showed that. That is another thing. Again, let's park it to the left side. Third thing which we talked about was the margins being catered to, or the margins being enjoyed by the distributor or the chemist. No, this is not the thing. Most of the places where such products are given are not in the tier 1 cities. They come in the tier 3 cities. The tier 2 cities, like I'm talking about Maharashtra here, or I'm talking about all over India, none of our cities come in tier 1, tier 3 cities. So when we are talking about the tier 3 cities over here, there the generic and substitution market, which is more of a chemist based, would make a difference. In India, still about 1,42,000 crore worth of markets, 1 lakh crores worth of market is still in the branded thing. And whereas only 40,000 crores come from the generic side. So we are talking about different segments over here, and the issues which we park to the left require a full big session with the concerned industry experts to be taken into the limelight. But what we forget here is that India is a country which is so very proactive that the drug remdesivir currently being available for COVID patients, when it was launched by Gilead just 6 months back, was made freely available in India in just a short time. The first company in India to manufacture was Hetero, which priced its drug at about 5,400 rupees MRP. And now we have a company like Cipla selling it at 4,000 rupees and a company like Cadila coming at 2,800 rupees in just less than 4 months. So this was exactly what Umesh sir was talking about, the price compatibility and the time span in just getting. So the time span was not even months, time span was only days. And the proactiveness of the Indian pharmaceutical industry, which helped each and every patient get remdesivir, has been phenomenal. There are now close to 9 companies like I think Mylan, like Jubilant, like Cipla, like BDR, like Hetero, and so many. So all of these are the companies which are parallelly doing it and are in turn doing a great service. So Indian markets, till the time we have I would still say that the Indian pharmaceutical market is India un-Inc. It is not India Inc. Inc is INC Incorporated. So according to me, I still rate India's market as India un-Inc. I would like to call that term un-Inc. Because India is still greatest market. We see that Sun Pharmas, we see Abbott, we see Ciplas, we see Glaxo, and we see so many multi-crore figures. But India, when it is growing and India, when it is reaching to the pharmaceutical market, we are not dependent on the 5,000 crore companies in the 5,000 crore companies, we are looking at 1,005 crore companies. It's a different ball game altogether. So that is about the Indian pharmaceutical industry. Just a small thing. So sorry.
Thank you, Sangram.
It was a good discussion. So many points came out. And
India has
different
branded
perspectives.
India has branded generics. That's what it is.
Yeah, India has branded generics.
Branded generics. We don't have that here. We have brands and generics.
So I told
there
you,
are only
India and
two
US are chalk
classes
and
of
cheese.
molecules.
See, cetirizine of Cipla, branded you check and branded generic you check. There is hardly any difference between the two. Actually, the companies are taking advantage by putting it into the generic frame. That's it.
In India, before the law is made, the loophole is made.
Yeah. Jugad is our specialty.
General Manager of pharma company, Sanjay Oswal.
It is our honorable Prime Minister's birthday today. Let us not go into bashing him today.
Yeah. Any other queries?
Thank you.
So, wonderful presentation, Doctor. It's my habit, If doctor just comes to my... it's wonderful to have
it is his expertise
Umesh.
and his knowledge which makes you say doctor again and again.
Yes, he is more than doctor.
Correct.
Yeah, he's a more than doctor. We can call him PDF.
Just call me Umesh.
Yeah, whatever. Don't call him compounder.
He is Umesh, then also he is Umesh now also, and he'll remain Umesh, irrespective of what he is as a core person. So wonderful, Umesh, having you here, and you opening up the new thought process for generic drugs. It is our honor to present you a certificate, that also in the virtual mode. So let me do that.
I'd like to invite him in future when this pandemic is over. I request Umesh, Sangram from DIPSAR, even Mr. Sanjay Oswal, General Manager, I think he was there.
Sir, first accept the bouquet of flowers.
Thank you so much.
Which will be followed by the certificate. Let me just check the certificate. I am also not very well versed with the, the technology. I am learning it.
Umesh and Sudhakar are so popular among our classmates. that his friends from our classmates from the Dubai also commenting on the lectures.
Oh, great.
Yeah.
You know, we were a great class, all of us. If I look back, I don't see that in the new generation of students which we hire. You know.
What you have mentioned is that quality consciousness is very much needed as far as FDAs and other things are concerned. So it is our duty as a faculty members of the institutions that we mold our students in such a way that
That's critical.
ethical practices are followed. Yeah. So your certificate is here.
Here
Yeah,
also it is written Dr. Umesh Pai.
Yeah.
I will correct it before sending.
Yeah, please. Yeah, thank you. Thank you so much, all of you, for the opportunity. In fact, it's a great honor to speak in front of all professors and teachers. Thank you, Professor Wakode.
Thank you, Sharad, for you.
Yeah. So
Thank you, everyone.
Yeah. Thank you. Have a good day.
Yeah. So all the participants, the delegates, the fourth day session closes over here. We will meet tomorrow at 9:50 a.m. and we will have the fifth day, happening fifth day like the previous 4 days. And we look forward to meet you tomorrow. Have a good evening and good day.
Thank you, madam. Thank you.
Thank you, ma'am.
Thank you, Sangram.
Thank you, madam. Thank you, sir. Thank you very much.
Thank you, ma'am.
Thank you, Sanjay. All the delegates for patiently listening and being here for long.
Thank you, Sharad. Thank you very much. Very good insights.
Thank you. Thank you.
Thank you, sir.